Thank you, Owen. Thank you very much for the kind words, and thanks to all of you for coming out this evening to hear a bit of a whirlwind tour of what we've been doing at the University of Guelph. It's a wonderful opportunity for me to be here in a community that values research and have colleagues like Owen and others at the university who are passionate about helping to get the word out, because I hope, as you will realize during part of this presentation, a lot of what happens with the science that we develop depends on you and what you and civil society decide we can do with this technology, and what we urge our regulators to do with it, and how we hope that it will change and improve life, which of course is our tagline at the University of Guelph. But before we get into all of that, all good stories have to have actors. And our story on biodiversity starts arguably with Carl Linnaeus and a lesser known figure, Petraeus Artidi. These gentlemen, about 250 years ago, came up with the idea of having a lingua franca for science to describe biodiversity. In other words, having a genus and species name and a formal description of each species on the planet. So in Linnaeus' time, he undertook his landmark volume Systema Naturae to begin cataloging species and writing descriptions of them for the greater glory of God. We thought at that time that species were unique creations, but 100 years later it was Darwin, with his thesis of descent with modification by natural selection, who gave us a mechanism for understanding the existence of species. Species exist because they evolve. And this is part of the backdrop that helps us understand why telling species apart can actually be tricky business. Some of the things that we commonly encounter, yeah, sure, a dog and a cat look very different from each other. But what about a dog and a fox? Or a fox and a dingo? Or an Arctic fox versus a gray fox? Where do we draw the line, and what's a species? It's not always easy. And we've been describing species in this way using traditional methods, looking at differences in shape and size and color and behavior and geographic distribution as kind of proxies for reproductive isolation. And during that time, we've described about 2 million species. But we don't even know how many species we share this planet with, not even to an order of magnitude. Some researchers have suggested it could be as few as 8 to 10 million species. But when you talk to folks who study bacteria and protists, they say there are probably 50 million species of protists alone. So our methods of trying to describe biodiversity based on traditional morphological traits has given us a significant understanding, but still incomplete. We've literally mapped more stars in the sky than we've named species on planet Earth. And this is one of the grand challenges of biodiversity science, is to try and enumerate the diversity on our planet. Another challenge that we face is, against the backdrop of this traditional knowledge of diversity, our keys that we would use to identify a specimen, say our Peterson Field Guide to Birds that we might use to identify a bird at our feeder, typically written based on differences of adult, intact, often male specimens. The problem we face is that biological specimens come in many forms. Things like eggs and juvenile stages, or even processed products where the morphology has been stripped away. So even for the species that we have described and have some understanding of, it can be very hard to apply that taxonomy to real-world cases where we might want to be able to ID larvae or fragmentary remains or juvenile stages. Fortunately, DNA-based identification works on all life forms. The reason is because there's DNA in every cell of every organism. Species exist because they evolve. One of the things that you need to understand is that DNA, the recipe for all of the proteins in our bodies, what makes us us, is a language of just 4 letters: adenine, guanine, cytosine, thymine. And when we sequence that DNA, we can use these different color bars as representations of the different base pairs. And at a molecular level, over time, mutations accumulate in different lineages. So we can start to see different mutations in different species. So using this DNA-based approach to identify species, we can go in and look at a short standard marker gene and use it like a DNA fingerprint, in this case a DNA barcode, to tell species apart. So you can see here between sockeye and Chinook salmon there are a number of base differences. And those combinatorial differences in their DNA sequence can be used to identify them, much like a barcode. That was a metaphor that was coined by my colleague Paul Hébert, who is the founder of our Biodiversity Institute of Ontario in this landmark paper of biological identifications through DNA barcodes. He recognized that this is a scalable approach because we can automate DNA-based identifications. And so if you're doing a traditional taxonomic survey, trying to understand the butterflies or insects in the dairy bush, you might need a number of different taxonomists who could key out mayflies versus beetles versus bees. The specialist expertise required to do biological identifications with traditional tools just doesn't scale very well. But if we could build a database that would house barcode profiles for all the species on Earth, we could build a digital identification system for all of life. That's what we do at our Biodiversity Institute of Ontario. We're using high throughput liquid handling robotic workstations that have come out of projects like the Human Genome Project. And we are the headquarters for the International Barcode of Life project. This is an international program that involves thousands of researchers around the world working to build a DNA-based identification system for all of the plants and animals on Earth around us. So the DNA barcoding process is done in a laboratory, like the Biodiversity Institute of Ontario, where we'd take a bit of tissue, extract DNA from it, PCR amplify that barcode gene. That gene has got desirable properties in that it has very little variation within species, but deep divergences between them. So we can use it to tell species apart. We then sequence that barcode segment and put it in a database. PCR sounds complicated, but it's really pretty straightforward. We're just Xerox copying that little gene region, and as we do so, we're adding different colored fluorophores to the 4 different bases so that we can run it on a sequencer and reveal that sequence of GATC. So it's just a simple chemical amplification step that allows us to go from taking a bit of tissue to reading its DNA sequence. But this process can take days in a lab. And as we've been doing this work and building barcode libraries now for more than a decade, involving researchers from around the world, started to see things like the UN Biodiversity Strategic Plan recognizing DNA barcoding as a key element of how we manage and monitor biodiversity globally. So this is bringing a lot of recognition to the university and to Guelph, but it's also being seen as a very important tool for conserving life on Earth. So DNA barcoding requires a reference library of known species. A lot of the work we do as researchers is to work with museums and herbaria around the world to get expert identified reference specimens and genetically profile them. And that will allow us to build a master key that can ID all life stages and provide these IDs quickly and cheaply with relatively little taxonomic uncertainty. And I just want to depart for a second to talk about an interesting case where the way we use barcoding to identify species has both inflated and decreased the number of species that we've seen in the world. In some cases, what we thought were just a single species of little silverfish have many deeply divergent lineages. Their barcode sequences are very different. So superficially, we might look at that and say, oh, it's one species. But then you look at the genetics and you say, there's something going on here. You know, to a 2-meter tall, visually focused vertebrate, just looking at it, I don't see difference. But they see differences between each other. We would call that cryptic species. So in some cases, we think of we're dealing with a single species, but the DNA shows that they're actually 2, sometimes more. In other cases, like this deep sea fish, we've named it as 2 different species in 2 different families. Now, these things come from the abyss. You would never see them in copulo. So it was natural that as they were hauled up in dredge samples that they got different names because they look different. But in fact, the barcodes are identical. And what we now know is that it's a male and female. they're sexually dimorphic. These two life stages don't look anything alike. So using traditional tools of morphology, we've erroneously ascribed them as being in two different species. In fact, this case, three different species, because the larvae look nothing like the adult males or females. So they were named three different species in three different families, and the barcodes helped us recognize that in fact it's one species with a variable life history. So this tool, geneticists and taxonomists are working together to kind of refine our understanding of biodiversity and develop a library or a catalog of all of this information. So we have the Barcode of Life data system, which is the information system that we pull all of this data into. We have a workflow where as I mentioned, we're working with collections like the Canadian Museum of Nature, the Smithsonian, the Royal Ontario Museum, and many others to access specimens that have been identified by experts, many times even type specimens. We put all of the data about where they were collected, GPS coordinates if we have them, who collected them, when, along with digital photographs into the database, and then we go through that barcoding workflow and we put the sequence in. So that couplet of a species name and a sequence becomes our lookup table. So as we build this reference library for known species and occasionally discover some new ones, we get to a point where we can use it in some interesting applications. My role over the past decade has been to help coordinate the global fish Barcode of Life campaign. So I've been working with ichthyologists from around the world to build barcode reference sequence libraries for fishes. Today we've barcoded more than 100,000 specimens representing more than 10,000 species of fishes. So we're not done yet. There are quite a few more to go, but we've gotten the vast majority of things that humanity comes into regular contact with in the database. And we're using it in some interesting ways. I could spend probably a whole semester lecturing to you about all the different applications that we found for barcoding. I'm going to try to cover just a couple tonight and then allow my colleague, Dr. Naum, to describe how she's taken this research even further to address some of the limitations of traditional barcoding. So when we're trying to identify things that can't be easily identified, we found in some traditional Chinese medicine shops, for example, things being sold as shark fin that actually turn out to be whale shark that's a critically endangered species. So we're using this tool to identify black market trade in threatened species. We've also done some work with fisheries managers as they're trying to restore spawning habitat for things like lake sturgeon or walleye here in Simcoe and in the area. So they try to undertake strategies to improve the spawning habitat for these fish, and then they go out and they can collect larval fish, and they'd like to know, are the species like lake sturgeon and walleye that we're trying to restore actually using these habitats? Because it can be notoriously difficult to accurately identify egg and larval fishes to species. So we're using DNA barcoding to help them understand the efficacy of some of these restoration efforts. I won't say much more about those two examples, go in a little bit more depth about some of the biological research that students are doing with this barcoding in the lab, and a little bit on some of the food safety work that we've done. So former PhD student Tim Bartley was very interested in understanding the food web structure in Canadian shield lakes. So when we look at fishes and other predators in a lake, you have what we call a food web, different species eating each other. There was some view that offshore species and nearshore species didn't really interact, that there wasn't much coupling. His question was interested more in nutrient flows. And are these two different, more the littoral zone versus nearshore, connected through these food webs? So he worked in part of a larger study that was collecting specimens from various predators in different boreal shield lakes. And one of the questions was really driven by the idea of, what if we introduce a non-native species like smallmouth bass to some of our iconic lakes? Does that change the food web structure? Does it upset the trophic dynamics of some of our native species like lake trout? So we were comparing lakes with and without introduced non-native species, trying to reconstruct food webs. And the challenge you have when you look at the stomach contents of a fish is it's stuff like this, little bits of a vertebral column. You can say, well, it's vertebrae, but How do you say what species is in the stomach of another fish? Tim went around collecting fish and barfing them and sitting and looking through a microscope trying to identify stomach contents the old-fashioned way. And it was pretty hard. But what was really exciting as we began to apply DNA barcoding, we could take a bit of that little vertebral column, extract DNA, amplify, sequence it, compare it to the database and say, oh, yellow perch. Lake trout are eating perch. That's fascinating. And they're eating dragonflies, and they're eating frogs, crayfish, smallmouth bass. You could even identify some of the parasites like tapeworms in the stomach. So what we began to see is that the resolution we could get using morphology of these degraded gut contents versus DNA barcoding, so we're getting a lot more fine-grained information about who's eating whom. And so that really started to increase the resolution of our food webs. The morphology gave us a partial picture of which species were eating which, but DNA barcoding showed it was a lot more complex. And that kind of flew in the face of conventional wisdom, because when people had observed these fishes feeding, they'd say, well, you know, lake trout predominantly feed on these minnows, but they don't go inshore and feed on insects. Well, actually they do. There's a lot of movement back and forth between near shore and offshore. So what we began to learn was that there's far more coupling between these environments. These food webs are far more dynamic. And instead of being very narrow and a few common prey species, we see that actually there's a huge amount of dynamic interaction there. So from a basic research standpoint, we learned quite a lot. And it was an interesting thesis for Tim. But just wanted to give you a flavor of some of the kind of applications that we can do with that. You may have seen in the news that Canada recently received a large grant from the Canada First Research Excellence Fund called Food from Thought. We are Canada's food university, and as Owen mentioned, we've done some interesting work documenting food fraud. So beyond some of the basic science that we do, understanding food webs, natural history, ecology of species using barcoding, we can also apply it to things like market surveys. As we started to build this barcode library for fishes, one of the questions that arose in the mind of myself and a former graduate student, Eugene Wong, was, is our database mature enough that if we just went out into the stores and started to buy seafood samples, could we put a species ID on them? So we started doing some market surveys, and work that Amanda has continued as well and played a very large part in. We would ask the question, does the scientific name that we get from barcoding match the market name that was listed on the package? Canadian Food Inspection Agency has a list of what species can be called what common name. We used that regulatory framework to determine whether or not a sample was mislabeled. So we went and we bought market samples, in this case from New York City and Toronto, barcoded them, identified them, and we weren't really prepared for what we found. We were pleased in that we could put a species level identification on everything we tested, even back in 2007, but shocked to find that 1 in 4 fillets was mislabeled. Always a species of a lower economic value being substituted for one of a higher economic value, which seems to suggest economically motivated adulteration or food fraud. And it got a lot of press. We repeated the study, and some original concerns were, well, okay, you did this one little study, but does that hold up? Well, we started working with reporters from the Toronto Star and the Vancouver Sun and Marketplace and others, and collecting samples from all across Canada. And we found that it doesn't matter if you're in BC or the Maritimes. This seafood substitution was systemic in the industry across Canada. And his colleagues have been repeating these studies in the US and Europe and South Africa and elsewhere. We find that it's systemic in the industry globally. That's unfortunate for a number of reasons. Economically, consumers should get what they pay for. Fraud is illegal. Environmentally, we see that sometimes overexploited species are being marketed as something else. So we may be unwittingly contributing to the decimation of some collapsing fish stocks through a process called illegal, unregulated, or unreported fishing, where species that aren't supposed to be being marketed are laundered as other things. So it has not only economic but environmental consequences, and importantly, we've also seen some human health consequences. We've seen things like puffer fish that have tetrodotoxin that can be poisonous, mislabeled as monkfish, and other instances where escolar can be mislabeled as tuna. Now, eating escolar isn't going to kill you, but if you eat more than a couple of ounces, it will give you a nasty, oily diarrhea, which probably isn't something you expect to get when you buy a nice sushi dinner at a restaurant. We've also seen some other species that can bioaccumulate mercury being mislabeled as other species. So there are environmental, economic, and potentially human health consequences to this kind of fraud. One of the most common seafood substitutes we've seen in this imposter syndrome, Asian catfish. They're farmed. A lot of the mangrove swamps and places in Southeast Asia are being converted to farm this particular catfish. And it's turned up in markets around the world, particularly in North America, masquerading as anything from flounder to cod to halibut to plaice to sole to grouper. It's amazing to think that this kind of substitution can happen. This is a very cheap farmed species, quite often being mislabeled as wild. We see a lot of cases where other farmed fish, farmed Atlantic salmon, are being passed off as wild Pacific salmon. So one of the things that I wanted to get up on a little bit of a soapbox and talk about is part of the problem is the fraud. People are cheating. Part of the problem is also our rather lax regulatory framework. So Sea Choice issued a report card earlier this year on international seafood labeling requirements. The European Union has started requiring things like scientific name, production method, harvest method, geographic origin, country of last major transformation or processing. So EU is actually doing quite well on their labeling, and through a combination of regulatory testing and consumer awareness, their rates of seafood fraud have gone down. The US and Canada not faring quite so well. More recently, the US Food and Drug Administration has started to use DNA barcoding to police this kind of seafood fraud, and I'm pleased to say that we've been collaborating with the CFIA over the last 2 years to start to introduce this tool in our own regulatory framework. But I think we also, in addition to doing this kind of testing and policing the supply chain, we also need to be keeping pace with updating our regulations and giving consumers more knowledge about what it is that we're actually buying when we go to the market. I could go on at great length about that, but just suffice it to say that right now we have different common names like sole, where actually 21 different species can be legally marketed as sole in Canada. After years of testing, only 4 of those species that can legally be called sole have ever turned up in our market testing. We found a lot of other things like that Southeast Asian catfish that is not supposed to be called sole, being sold as sole. But this is kind of a problem because it breeds the illusion that, well, my grandmother cooked sole, my mother made sole, I can go to the grocery store and buy sole. It must not be in decline, right? It's a plentiful fish. It's been around for a long time. Well, what I suspect has happened, given that 21 species can legally be called sole, but the vast majority of them we've never encountered in the marketplace, we've eaten our way through a number of different species. And as one stock collapses, industry petitions government to say, well, here's another flaky white-fleshed fish. Can we call it sole? The government says, sure. So we're unwittingly contributing to eating our way through the food chain and a number of different species in the process. So I like what the Europeans have done in starting to include a scientific name along with these common names so that we actually can know what we're eating. And also makes it a lot easier for us when we're doing DNA barcoding studies to say it either is or isn't that species. Because the species that can be called sole in Canada only partially overlap with the species that can be called sole in the United States. And then we find certain species can be called catfish here, again, only partial overlap with the US. In the UK, anything in 5 different families can be called catfish. So these common market names contribute to some of the fraud as well, this kind of quasi-legal market substitution in addition to the out-and-out food fraud. So When we do these kinds of tests, we're using a technique called Sanger sequencing to generate a full-length DNA barcode that's about 650 base pairs. It's a very small part of the overall genome of an organism, but it's enough to tell species apart. But it does have some limitations. When we're dealing with things like fresh fish or even baked fish or dried fish, we can get a barcode out of it. But when you start to tin things, that combination of high temperature and pressure fragments the DNA. So we can't actually get a barcode out of some highly degraded, highly processed food products using our traditional methodologies. Moreover, this Sanger sequencing approach works great when we're trying to identify a discrete fillet. We get a single barcode for that species, but if we've got a mixture of different species, we get their barcodes superimposed on top of each other, and so it doesn't work very well for mixtures, and it doesn't work very well for highly degraded DNA. But the research that Amanda's been doing is designed to address that. And I will stop now and let her tell you about what she's been doing in that space. Thank you.